So, hi again, and welcome everyone to the new session of the BioClock Academy series. Hope you're having already had a good day, and now it's only going to get better with a nice presentation. So today's session is hosted by me, Laura Pape, a PhD student at the BioClock Consortium, and Georgiana Roos-Oswald, who's a postdoc at the BioClock Consortium. And just a quick recap for those who don't know it yet. So the BioClock Consortium is a Dutch research organization. It was funded by the Dutch government. We just talked about the funding. To set up to study different aspects of the biological clock in healthcare, in society, and also in environment. And so we set up this BioClock Academy series to introduce you to basic concepts and equip you with knowledge surrounding chronobiology topics from expert speakers in the field. So that's why we're here. We have a 40-minute talk followed by a 15-minute discussion. If you hear the talk and you think, oh, I have an important question, just write it in the chat already, and then we will read it out loud, or afterwards you can unmute yourself. Yes, and now I give the floor to Georgie, who will introduce our guest speaker for today.
Hello, everyone. Thank you, Laura. Welcome also from my side. I'm very happy to introduce you to our guest speaker today, which is Mariana Astiz Cadenas. I hope I pronounce it well. I try to keep my Spanish back, but I think the region where she comes from, there's a bit of a special Spanish because she comes from the Achucarro Basque Center of Neuroscience in Bilbao. So it's located in Leioa, which is the university campus there. And she is a leading researcher in circadian biology and neurodevelopment, and her research is mainly focused on the biological clock and how this is shaped in early life. For example, she also looks at and explores the impact of certain hormones which are used in the prenatal medicine, like the antenatal glucocorticoids, and on the development of the circadian system there. So she combines neuroscience, endocrinology, and chronobiology, and her research provides really valuable insights into how early life exposure influences long-term health. But I don't want to spoil too much of this, and I want to give the floor to Professor Cadenas, and I'm very looking forward for the talk. Thank you very much for being here and bringing us some Spanish vibes.
Okay, thanks for the invitation. Welcome, everyone, to this talk. It's a pleasure to be here online for now, but then soon in person. So yeah, it's a pleasure for me to be here. So I'm a Spanish speaker. This is my mother tongue, but actually I'm from Argentina, from the very south of the continent. And then, well, I moved during my career in many places, including what you will see, so the longest postdoc I've done in Germany. And then I'm here in the Basque Center for Neuroscience, trying to answer this question that I'm posting here: when and how the circadian clock starts to tick. Yeah, and so here. So here I brought just a little map for you to see where Bilbao is. So this is Spain, the north of Spain, very close to it's very close to the sea. And this is a lovely view of Bilbao. One of these days that it's sunny, like today, but this is also not the normal situation here as well. And here is the group of people. This is the latest picture of the group. The people who really are motivated to join and to try to answer this complicated question. So today I would like to focus this question on some studies that we have done on the development of the circadian system, and specifically on the role of antenatal glucocorticoids. So just as a brief introduction, I would like to put only my timer so I'm not over time, because I would like to have your questions. So I got interested in the circadian system around like 12 years ago when I found out that it was a really cool system to study, and that it's basically present in almost every organism on Earth, from humans to cyanobacteria to plants. And this is because it's an adaptation mechanism that we have developed by living in planet Earth, that it's rotating around its axis every 24 hours, and by that changing daily the light-dark schedule. And what I found interesting about circadian system is that it can anticipate these changes, and by that optimizing behavior, physiology, and every cellular process. And this is exactly what it happens when we can anticipate the problem. Of course, we have more time to think about the solution. And I think this is, and the fact that it's present in so many different species speaks about something really important, at least to me, and something that is worth studying. And over the years, this is, as you know, not a super old field of research. And over the years, people have seen circadian regulation on behavior, on different physiological mechanisms, and also different cellular processes at the mechanistic level, at the molecular level. So, how does it work? So I also find interesting the organization and the way in which the system is organized. So this is basically some of what we know from studies in mice. So this is the brain of a mouse cut in a sagittal manner. And so we have, so the rhythms are 24-hour rhythms are organized in a hierarchical way with a master clock in a very tiny region of the hypothalamus. The hypothalamus is the main brain region that it's in contact with the environment and tells the rest of the brain information about the environment. And this is what the suprachiasmatic nuclei is doing. I would call it SCN for short. So in this case, this nuclei, it's connected to the retina through the retinohypothalamic tract, and this is how it receives light-dark information. It integrates this information with other signals, and it's able to send this information to other regions of the brain and organize the rhythmicity of the functions that these parts of the brains are performing. And also, it's able to communicate to other hypothalamic nuclei in order to organize the rhythms of hormones and the rhythm of the tone of the autonomic nervous system. With this mechanism, the SCN is able to tell every single clock in peripheral tissues also what time is it in an integrated way. So the whole body is in sync with the environment. So, but for this to happen, we need that the cells, almost all the cells, have a molecular clock. This means that the signals that are sent by the SCN has to be understood by the cells. And this is done by a molecular clock mechanism that works as a transcriptional translational feedback loop that oscillates every 24 hours and has as targets what we call clock control genes. So this makes the transcriptome of each cell oscillating in a 24-hour manner, and in this way, the functions of the cells can be synchronized in this way. So the main signal that we are interested in are glucocorticoids. Is one of these signals which rhythmicity is organized by the SCN and released to periphery. So this is basically a plot of a profile of glucocorticoids in a diurnal animal. So like us, for instance, we have our active phase during the light and our inactive phase during the dark. And then the peak of glucocorticoids for us happens in the early morning. Instead, for the animal models that some of the animals models we use in the lab, they are nocturnal, but the profile of glucocorticoids here is quite similar. They are active during the night, but the peak of glucocorticoids is still at the beginning of the active phase. So we know quite well how this works in adults, but a big question still in the field is how this whole system is organized during development. And this, as I said, it's the main question we have in the lab. So we do know some things. So we know that the circadian system is gaining function perinatally. there is a slightly difference between different species, whether it's a little bit before birth and after birth, or completely before birth. But in all the cases, there is a critical window in which the early environment in which we are exposed to defines how the circadian system works later on in life. And because the circadian system, as I told you before, is making us ready for the next time of day, it impacts, so this early environment impacts in our physiological fitness later on in life. So if we think about this critical window, if it's perinatal or prenatal, there will be, so in the case of pregnancy, it will be some maternal signals that will be rhythmic, like glucocorticoids, like melatonin, like dopamine, that will be able to cross the placenta and tell the baby or the baby clock that is developing what time is it. So 10 years back, I joined the lab of Henry Koster in the University of Lübeck in Germany, and we were wondering what would be the importance of the rhythmicity of these signals, whether only the signal was important or also the timing. And this was our main question back then. We wanted to know whether the time of antenatal glucocorticoid exposure can determine the long-term effects that have been shown by glucocorticoids perinatally itself. So basically, we designed a very simple paradigm. We had mice that were pregnant, and we inject them with corticosterone, which is the main glucocorticoid in mice. And we choose, so here is again the plot that I show you. The peak in mice, because they are nocturnal, is right before the active phase in the dark. And so we wanted to check this time idea. And we simply inject a group of mice in phase, so right before the active phase. And we inject a group of pregnant mice out of phase, right at the beginning of the inactive phase. In this way, we had like these two groups, out of phase or in phase, and we did this intervention during the last part of pregnancy, in which most of the hypothalamus and also the circadian system is being formed. So at the end, we let the mice be born and grow, and we analyze the behavior of the offspring. And we compare basically those offspring whose mothers were injected out of phase with this offspring whose mothers were injected in phase, with a naive group that was not perturbed during the whole pregnancy. So we analyzed a battery of behavioral tests to see whether we can see any difference by the timing of injection. So the first thing we did is to measure the circadian activity. As I told you, these animals are nocturnal, and this is plotting locomotor activity in counts per minute across the day. And as you can see here, you have more activity of the animals during the dark phase and really little during the light phase. So, but here, when we compare the groups, we start seeing the first main difference. So this gray group, which is the offspring of those mothers injected out of phase, they were running less during the dark phase, and they were running more during the light phase when they are supposed to be sleeping mostly. And this activity in the light phase is quantified here. We didn't have a good explanation for this. We said, oh, okay, maybe they are hyperactive, maybe they have some kind of a sleep disruption. But it was a good observation already that the timing of corticosterone was doing something to these mice. Then we said, okay, let's go for other stress-related behaviors like anxiety-like behavior. We use the elevated plasmase for this, and we measure the number of open arm entries. This indicates how anxious the animal could be. The less times it goes in the open arms, then the more anxious the animal is. And as you can see here, compared to the naive group, we have a higher level of anxiety in those whose mothers were injected with corticosterone, but also we had a bigger effect on the out-of-phase group. Same happened when we measure stress coping behavior. This you can test with a test that is called force swim test. And here what you measure is the time in which the animal stays immobile. And here again, we saw that those animals whose mothers were injected out of phase were less capable of coping with a stressful situation as being in the test. So in a way, this indicates that not the concentration, but the timing of injection was giving us a different behavior. And this behavior, because glucocorticoid is a stress hormone, this behavior could be related with the stress axis. So that's what we did. We tried to characterize everything that happened to the stress axis in these 3 groups of mice. So as you know, the stress axis is activated in the paraventricular nuclei of the hypothalamus. It releases CRH and ABP to the pituitary gland. The pituitary releases ACTH to periphery, and the adrenal is the one that is producing glucocorticoids that goes to target tissues. Glucocorticoids signals back the PVN and the pituitary to shut down the system. So the system is reactive anytime that the stress there is some stress in the environment. So to analyze all this, the first thing we did is to measure the glucocorticoid or the corticosterone levels during the rest phase. So as you can see here in the out of phase group and the in phase group, we have higher levels of glucocorticoids during the resting phase of the mice. This indicates that somehow the HP axis is kind of constantly activated. When it's constantly activated, the problem that we have is that when we need it to be activated acutely in front of an acute stress, it's not so efficiently activated. And this is exactly what we see when we induce an acute stress paradigm to the mice. So if we measure the fold change of glucocorticoids before and after stress, we see that the naive group increased the glucocorticoids almost 8 times, while the other 2 groups increased the glucocorticoids, but not so efficiently, especially those whose mothers were injected out of phase. And so then we said, oh, okay, this might be because there might be a problem with the negative feedback mechanism to the PVN or the pituitary. So we did another experiment to test this. And this is basically an injection with dexamethasone that you do 6 hours before when the peak of glucocorticoids, physiologic peak has to happen. And then you measure how the HPA axis is suppressed by dexamethasone, which is an agonist of glucocorticoid receptor. So what you expect is what you see in the naive group, that by injecting dexamethasone before, then you have an inhibition or suppression of the activation of the axis, and this doesn't happen in the out-of-phase group. So we say, okay, then it might be an impaired negative feedback mechanism. So this means that something can happen at the level of the PVN, and we wanted to check the expression of the glucocorticoid receptor in the PVN because it's there where the system shuts down. And what we observed comparing these 2 groups is that the out-of-phase group had expressed lower levels of glucocorticoid receptor, indicating that even if there is glucocorticoids around, it cannot receive the signal as efficient. And we also said, okay, but then this also it's not only the reception of the signal, but also how sensitive the glucocorticoid receptor is. And we test this sensitivity of the glucocorticoid receptor in periphery with a very simple ex vivo experiment. So we had a full blood and of these mice, and we incubated with LPS. So LPS is the external cover of gram-negative bacteria. So we induce a pro-inflammatory response, which can be inhibited by glucocorticoid signaling. So that's why we use LPS alone on LPS and dexamethasone, and we measure a pro-inflammatory cytokine as a readout of the pro-inflammatory reaction. So as you can see here, so the sample of these mice had LPS alone or LPS with increasing concentrations of dexamethasone. So dexamethasone for the in-phase group was acting nicely and inhibiting the production of IL-6. But in the out-of-phase group, we also see that we had to add more concentration of dexamethasone to inhibit IL-6 production to the same level. This means that somehow the sensitivity of the glucocorticoid receptor was impaired in these mice. So with all this, we said, okay, so there is something that we are doing or something that is happening during the injection time that is what is giving us this differential setup of the HPA axis and this differential behavior. So, and we call this mechanism a gating, meaning that the glucocorticoids have one effect at one time point and they don't have this effect at another time point. So we wanted to understand this gating mechanism. So we went back to pregnancy and we had again the 3 groups. But now instead of analyzing the offspring, we analyzed the fetus in order to understand which clock was mediating this gating. Because we had 3 options. We had the maternal clock, so you can inject the mother, and the mother can handle differently depending on the time of day, the amount of glucocorticoids that we were giving. The placenta has an enzymatic barrier for glucocorticoids, so it could be that the placenta clock was filtering differently depending on the time of day, or it could be that it was the fetal clock, which can be gating the reception of the signals differently. So the simplest idea we had to test this is to test the amount of corticosterone that was in the fetal blood right after the injection. So here is the values in the naive group, so a little bit higher during the dark phase. And this is the values that we have 1 hour after injecting, either in the morning, so out of phase, or in the evening, in phase. And as you can see here, we have a rise, like an important rise in glucocorticoids 1 hour after injection, but there is no significant difference in the level depending on the time of injection. So this let us think that maybe it wasn't the maternal clock balancing differently the levels of glucocorticoids that we inject, and also not the placental clock filtering differently. And then we focus on the fetal clock. So to do so, we thought, okay, we need to focus on a tissue that is highly receptive to glucocorticoids, so express high levels of glucocorticoid receptor at this particular time point, and something that explains us the behavior on the HPA axis that we saw before. So we chose the hypothalamus, and we isolate hypothalamus of this fetus. And we analyze there the expression of a glucocorticoid receptor target gene. So this target gene will be induced if glucocorticoid receptor gets activated by glucocorticoids. So this is the case of FKBP5. So here in the dot line, we have the controls without injection. And we see here the fold induction of this glucocorticoid receptor target gene 1 hour after injecting glucocorticoids to the mother. And as you can see, when we inject out of phase, then we have a rise over the controls, but we don't see this when we inject in phase. In fact, we see a downregulation of the gene. So this let us confirm our idea that maybe the fetal clock was already ticking and that maybe it could gate the activation of the glucocorticoid receptor. And then that would explain why the same amount of corticosterone that we inject to the mother program prenatally differently, and we see a different stress response in the offspring. So in order to check this, we had to remove the clock from the fetus. So we did so by mating BMAL1 heterozygous mothers with BMAL1 heterozygous fathers and injecting with corticosterone either in phase or out of phase. In this way, from the fetus that we got, we had those injected out of phase that contained the clock genetically or were clockless, or those injected in phase with clock or without clock. And we did the same. We analyzed the expression of glucocorticoid receptor target gene. So first we compare with the control here in the dotted line, those animals who have the clock, like the wild types that I showed you before. And again, here we saw an upregulation over the controls when we inject out of phase. What happened when we remove the clock? When we remove the clock, then the out-of-phase injection induces the expression of this target gene, but also the in-phase injection. So this indicates us that the fetal clock was necessary to be present there to gate the activation of the glucocorticoid receptor. So, the answer whether the time of antenatal glucocorticoids really determine the long-term effect is a yes for now, but we have still a lot to do. Now we propose that the gating mechanism is controlled by the fetal clock, and that's why the timing of maternal corticosterone exposure determines the behavior. But now we have a lot of more questions, as always happens in science. So how is this gating mechanism controlled at the molecular level? Who is controlled? It's the glucocorticoid receptor level. It's the binding of the receptor. It's the binding to the DNA. We are working on this. Katerina, it's a master student working on this. Xavier, it's also a PhD student from my lab working on this at the moment, more at the molecular level. But we also wanted to understand how this prenatal programming happened and how this lead to the altered behavior. And for this, we need to understand whether this is methylation of genes or like some epigenetic change that explains why this intervention during early life are maintained until the adulthood. And for this postdoc in the lab, Mariana, it's working on this, and we are working in collaboration with Henriette Kirchner, also in Lübeck, trying to find out where the methylations groups are. So I hope to tell you something more in the near future. So, but the other question that we have, so is this really interesting for someone else than for pure mice or experimental scientists? And we found out that this might be interesting because you know that the antenatal glucocorticoids is a therapy that is given to 90% of the mothers at risk of preterm delivery. This happens between gestational week 24 and 34. I think depends a bit on the hospital. But basically, these antenatal glucocorticoids are given to the mother to promote the maturation of the fetal lungs, because these antenatal glucocorticoids reduce the risk of the respiratory distress syndrome. And this is the main cause of mortality in premature babies. But it's also known for the last decade that this treatment with synthetic glucocorticoids increase the vulnerability to develop stress-related disorders later in life. And also something that for me was really striking is that it's so difficult to predict when a preterm delivery will actually happen that the glucocorticoids are given just in case, but then 30%, at least in the hospital that we have data about this, 30% of the cases, then the mother ends up delivering term. So this let us think that if this timing effect was also happening in humans, then we can keep the benefit of giving these antenatal glucocorticoids for lung maturation for the baby, but we can reduce the vulnerability to develop stress-related disorders later in life. So to try to start checking this, we started a collaboration with Christoph Hertel. So he's, he was back then here in Lübeck. So this is, he's part of the Sherman Neonatal Network. It's 68 different neonatal intensive care units in the whole Germany. And basically, it's a population-based multi-center cohort that enrolls very low birth weight babies. And then it follows them until 5 years old with a lot of different markers and samples and so on. So we said, okay, if we can link these babies or these kids that are being assessed in this network to the time of injection of the mother, and we can compare both, then if we assess behavior, we can know whether this might be also reproducible in humans or not. So we did that, and we made 2 groups of infants, ones whose mothers were injected in phase, now at the beginning of the morning, and some of them injected out of phase at the end of the evening. And then we studied a questionnaire in which the parents were asked for some behavioral features of these kids. And we chose those questions that were related to those behavioral tests that we did in mice to try to make a parallel. So when we analyze this, we create a behavioral score and we compare between the two groups. And here we also saw that those babies whose mothers were, or kids whose mothers were injected out of phase, had more of these features of hyperactivity, bad temper, anxiety, and they were stronger than those babies whose mothers were injected in phase. So this is a really small study, but we are now trying to increase the sample size in order to understand whether considering the time of antenatal glucocorticoid treatment could be really reducing the long-term consequence, or at least some of the long-term consequences of being born too early. And that's why we are also collaborating with a hospital here in the Basque Country to trying to see whether in different sites, in different countries, this is also true. And also we had the idea of trying to predict behavioral difference through molecular changes in babies, because this could allow us to do some kind of intervention on the babies or the kids' stress management. And I don't know if you remember when I show you this essay that we did in the mice, in which we test the sensitivity of the glucocorticoid receptor in periphery, in blood. We did the same in the babies. So, and here we see something very similar. In those babies whose mothers were injected in phase, the glucocorticoid receptor sensitivity seems to be okay, because the IL-6 production is reduced with higher concentrations of dexamethasone, but the out-of-phase group requires more dexamethasone in order to inhibit IL-6 production in the same manner. So this was, of course, very interesting. We still need to keep working on this, and the more samples and information that we have, the better for making a good claim, really proof with numbers of babies in different places. So then coming back to this slide that I showed you before, when we try to understand the role of rhythmic cues, we realized that they were actually important. The rhythmicity was actually important, but also that the baby clock played a role there, gating the sensitivity to these maternal cues. And this, it's what made us really even more focused to answering this question: when and how does the clock really start ticking? And so coming back to this drawing of the suprachiasmatic nuclei, so when we think about this question, when and how does the clock start ticking, it's very difficult, because one can say, okay, for me the clock is the molecular clock, yeah, the TTFL, the molecular mechanism. And some others can say, oh no, for me it's like the systemic organization of the clock. And some others could say, like, no, well, the master clock is the master clock, so it's the most important. Okay, all of them are important, but we have to focus and start from somewhere. And we start focusing on the master clock. So now this is a coronal section of a mouse brain, and this is the size of the suprachiasmatic nuclei. Here is super, super, super tiny. It's on top of the optic chiasm and connected to the retina, as I told you, and has these two kind of main regions: the core, which receives the light, and the shell, which is who's organizing the endogenous rhythmicity. And it's as any other hypothalamic nuclei that it's described until now, is extremely complex in the variety of neurons that are forming the circuit. So this circuit, it's built by 20,000 cells. From the neurons, 90% of them are GABAergic, and you say, oh, okay, this is easy, it's only GABAergic neurons, but they produce a lot of neuropeptides, more than 100. So the paracrine signaling within these cells is extremely strong. And then also for some time now, we know that astrocytes are important players in this circuit, in the timekeeping of this circuit. Okay, so how do we get such a complexity and such an robust clock in the suprachiasmatic nuclei. So as I told you before, this happens during the perinatal period. We go from a group of neuroprogenitors that are not really connected to each other. They don't know what they would be in the future. They are deciding their fate. And this happened in a very dynamic way during the perinatal period. And importantly, this process, that it's now a question mark, it's influenced by signals coming from the maternal environment or the early environment after birth. So our hypothesis is, it has two parts. From one side, we think that the sooner the rhythm stabilizes, it's the better for baby's physiology. And this is kind of coming out of logic to thinking that the circadian system at the end is optimizing physiology. But what we also want to hypothesize is that what is key is the circadian phase coherence. Because the clock itself, because this is also proven, it will oscillate with a low amplitude and will gain amplitude over time. And this will make the clock mature. And then also these signals coming from the mother can come at different times of day. And the idea is that if they come in phase, they can reinforce this endogenous oscillation. And if they come out of phase, as we saw for the glucocorticoids, they can somehow alter this gain of functionality of the clock. So again, we are interested in how this happens in humans and in mice. So we are collaborating with the Sherman Neonatal Network and with Christoph again. And the idea is to try to understand the timeline of development in human babies. So we had the idea of looking longitudinally different physiological parameters of these babies when they are born prematurely in the incubator, like body temperature, oxygen consumption, blood pressure, heart rate, and make out of it a kind of a circadian rhythmicity factor that we could correlate to gestational age. Of course, we here hypothesize that the older the baby is, the more rhythmic the system will be. And we also want to understand the influence of antenatal glucocorticoid timing. So is it true that when we give the babies glucocorticoids out of phase, we impair this correlation and we make the clock start oscillating with a high amplitude later? And when the injection is in phase, then we have a stronger clock. This is the question, one of the questions that we would like to ask, but we also want to include some more collaborators here, and this is when we were talking with Ines about this, and we will keep talking and trying to see whether we can amplify a network of hospitals or like researchers that we all interested in how this clock develops. So, and can we model the development of the master clock in mice? So in mice, as I said, is perinatal. And the first question, so we can, of course, model it, but we have to somehow start from somewhere. And so we ask one question that it's kind of recurrent question in the field, that is how the sensitivity of the SCN changes over time in development. Because what it's described so far is that early during development, the SCN is sensitive to glucocorticoids, but then something happens and it becomes resistant. So we wanted to understand this. Is it a downregulation? Is it a developmental program? How is driven? Who is in charge of this? So we did some single nuclei RNA-seq experiments. I will explain you now in collaboration with Malte Spielmann Lab in Lübeck as well and Violeta Pillars. So basically what we did is we obtained SCN only from 4 different time points. So 1 before birth in the mouse, and postnatal day 2, postnatal day 10 when the eyes actually open, and an adult sample. And then we analyze so we obtain the nuclei and we analyze the transcriptome of these cells. And we plot them all in this, what it's called a HUMA plot. So basically, in this HUMA plot, you have in 2D the a measurement of how similar or how different the transcriptome of each of the nuclei is. So each of these dots is a nuclei of a cell. And if they are close together, they are similar. The transcriptome is similar. And if they are far from each other, they are very different. So basically, here we put like the 4 time points together. And for our surprise, we saw that there was really little overlap between the cells that form the SCN at gestational day 17 and the ones that form the SCN at postnatal day 2, and a little bit more overlap in the cells that form the SCN postnatal day 2 or 30, 10 or 30. So this means that during this window, there is a lot of transcriptional changes happening. And so what we did next is to try to identify the type of cells that we have. And basically, we have mostly astrocytes, 1/3 of astrocytes, and basically neurons and other smaller population of cells. We identify them by marker genes that are known to be expressed in those cells. And then we said, okay, now we want to know what happens with the glucocorticoid receptor. So if we plot a glucocorticoid receptor expression, this is the name of the gene, over time, we observed that it was expressed in all the cells, in neurons and in astrocytes very early in life, but then it got enriched in this region of the cells that are astrocytes. There is also expression in some neurons, but it's much lower. So we wanted to validate this because we believe that the SCN is actually resistant to glucocorticoids in the adult. So we validate this by in situ hybridization, checking the presence of the glucocorticoid receptor mRNA and in astrocytes or in neurons. We found them in astrocytes indeed, and then we did immunohistochemistry to see at the protein level whether it was in the astrocytes, and we did find them in astrocytes as well. So, but what is doing in the astrocytes? So for that, we try to understand whether the glucocorticoid receptor in astrocytes in the SCN were responding to external glucocorticoids. So we chose, as I told you before, there is a difference between the morning and the evening corticosterone levels in a mouse. So it's higher in the evening because they are nocturnal animals. And then we simply measure the amount of glucocorticoid receptor positive cells in the nuclei in astrocytes. And we see that when we have low glucocorticoids, we have low glucocorticoid receptor in the nuclei, and we have high glucocorticoid receptor in the nuclei when the peripheral levels are high. Why are we looking in the nuclei? This is something I forgot to mention. This receptor is located normally in active state. It's located in the cytosol, but when the hormone comes, it gets activated and goes to the nuclei. That's why we are looking at the glucocorticoid receptor active in the nuclei. And so we said, oh, okay, but this could be related to the timing and not to corticosterone in periphery itself. So what we did is we took animals in the morning and then we inject either vehicle or glucocorticoids. And then 1 hour after, we measure the activation of glucocorticoid receptor in SCN astrocytes. As you can see here, this is staining for glucocorticoid receptor here. We have little when there are low glucocorticoids in periphery, and they are much higher concentration in the nuclei when we have high concentrations of glucocorticoids in periphery. So basically, this told us that along the SCN maturation, GR is not downregulated in every single cell, but it's downregulated mostly in neurons, but it stays being expressed in astrocytes. We also saw that in the adult SCN, the astrocytes do respond to peripheral glucocorticoids, either physiological levels or injected peripherally. So now we are working on trying to understand what does it mean for the SCN function and how can we relate the influence of glucocorticoids during the development of the clock with the function later on in life. And yeah, so with this, I hope I was clear enough. I would like to thank you for your attention and all the people from the lab that have been working super motivated to get all these results. And also Henry Koster and the people who collaborate in this work from the Institute of Neurobiology in Lübeck. And also the collaborators, of course, Christoph and Inmar are from the pediatrics department, and Malte, Christian, and Barun from the human genetics also in Lübeck, and also the people who trust us and give us funds for pursuing these ideas. So thank you very much.
Muchas gracias, Mariana. Thank you very much.